Okay, so again, we're back here. Here is our voltage-gated calcium channel embedded in our phospholipid membrane. This is going to be down here in the lower section because what we're trying to do is allow calcium that is outside of our cells in our interstitial fluid to go inside. And again, why are we doing that? We're doing that because this motor protein here that's pulling the vesicle, again, here's a vesicle, here's a vesicle. This is the vesicle that has the neurotransmitters. What we're trying to do is take this phospholipid membrane-bound vesicle and fuse it with the cell membrane of the outside of the cell, in this case, the cell being a neuron, so we can release the contents inside. And the contents inside is our neurotransmitter. So again, watching this guy actually walk. Whoop. Okay. This whole action requires, that whole action required both, and as that whole action of the motor protein pulling the vesicle with our neurotransmitter acetylcholine required calcium and ATP. And those are the two things that are required for a muscle contraction to take place. So we're going to look at that a little bit closer. Okay, so we're back here. Our action potential, that nerve impulse moves down. It changes the voltage here, causes this to open up. And now calcium is going to rush inside. Calcium rushes inside. Again, enabling our motor protein to pull the vesicles down and go through exocytosis. Okay, so we saw this calcium rushing in and triggering all the events that I've been talking about. And of course that causes the membranes of the vesicles to fuse with the membrane of the axon bulb here, and then causes us to release our neurotransmitter into our synaptic cleft. Here we see our neurotransmitter having been released through exocytosis to the synaptic cleft. And the reason the neurotransmitter is not going to go inside the muscle cell, but this motor end plate, this phospholipid bilayer, now instead of having voltage doorways in it, it's got what's called chemically gated channels inside of that. And what that means, that's doorways that need a key to open them up to allow ions to go inside and change the voltage on the inside. And of course, that changing of the voltage on the inside is called depolarization. Okay, so here we see an actual chemically gated channel, which I like to call a keyed doorway. It's a doorway because it allows things in. Here's the keyhole that opens up the doorway. Now,
potassium ions.
It's not allowing the neurotransmitter to come in. The neurotransmitter is attaching to it and opening up the doorway, which is allowing sodium to come in, and it's also allowing some potassium to leave, but there's always more sodium coming in than potassium leaving, and I'll explain that later. So therefore, since we're negatively charged on the inside, if I got more of a positive ion coming in than leaving, then eventually, if I have enough rush in, then I will depolarize that, and then for a short period of time, we would have that positive charge on the inside where that's happening. Now, right now, that's happening along the motor end plate, but whatever happens on the motor end plate changes the voltage on the inside of the cell, and then we actually start having voltage doorways open up that open up sequentially, and then more sodium keeps rushing in, and then as soon as sodium rushes in, potassium rushes back out through a different doorway in the sarcolemma, but it restores it back. In other words, it repolarizes it. Okay, here we see our neurotransmitter going to the keyhole, opening up the doorway. Next we would see sodium, a bunch of sodium rushing in, some potassium rushing out. When that happens, then we have just what we say, because we'd have these doorways spread all throughout this motor end plate, but we would not have these type of doorways throughout the rest of the membrane. They're only going to be found in the motor end plate, because that's really the only place where we're going to get the keys, the neurotransmitters, to open them up. Okay, now, when we depolarize that, then that's going to change the voltage on the inside, and then that change of the voltage will trigger the voltage gated channels to open up throughout the rest of the cell. But we don't want to keep those doors open forever. We actually at some point want to close those doorways, and we even want to, all that sodium that rushed in, we want to pump it out, and all that potassium rushed out, we want to pump it back in, and that's where our sodium potassium gates that we've mentioned before come in. But at this point here, we are depolarizing. Now, eventually though, I want to take the key away, so I have an enzyme in that's found in this region of the motor end plate and the synaptic cleft that has the ability to break down the acetylcholine into an acetyl group and a choline group. We've mentioned the acetyl group before when we talked about cellular respiration. Now remember, what are enzymes? They are proteins that either put stuff together, break stuff apart, or rearrange stuff, or at least it lowers the energy of activation so that can happen. And how can we tell it's an enzyme? Well, enzymes will normally end in ASE. The first part of the name will tell us what it's an enzyme for. So acetylcholine is going to be broken down by acetylcholinesterase. Okay, here we see the acetylcholinesterase broke down the acetylcholine into the two component parts, and then those parts are actually, and there's different ways neurotransmitters are reabsorbed. We'll talk about that later, but these are actually reassembled back in the neuron where they are recycled and reassembled into the vesicle so we can use them again. Okay, now again, once this motor end plate is depolarized, then all of a sudden our action potential starts moving along the sarcolemma down the T-tubules. And the reason is because even though they don't show it here, we have little voltage gated doorways that once we change the voltage inside, it causes them to open up. And once one doorway opens up, it changes the voltage for the next one to open up, which changes it for the next one to open up, which changes for the next one. So notice when we see the signal moves, it's not that everything becomes positive on the inside at one time, but it does so in segments because it is propagating from one area to another as we open up each of the doorways. Okay, so as the signal moves down, and again now this segment of that membrane, this is depolarizing. So that had depolarized, but now it's repolarized. It's gone back to the original state. It depolarizes again when sodium rushes in. It repolarizes when potassium rushes out. Now, the whole reason this signal runs down through this area is the same reason this signal ran down through this area. What was the reason the action potential ran from this area to this area? To open up calcium voltage channels so calcium could come inside. Well, the reason this signal is running down here is because the terminal cisternae of the sarcoplasmic reticulum of this triad here, the terminal cisternae also has calcium. We have to have a way to trigger it to be released into this area. So just like causing the calcium to come into this area triggered my motor protein to grab these vesicles and pull them, well, releasing calcium here is actually going to bind to troponin and shift tropomyosin threads off. So as long as we have ATP available, our cross bridges here will grab the binding site and pull them. Notice once the signal gets down here to where the sarcoplasmic reticulum is, then the voltage gated channels in there open up and release the calcium here into where the myofilament is and where the thick and thin filaments can now interact. Okay, one of my favorite things about this program is what it shows is what we were seeing was what normally happens in a normal muscle contraction. But what if we have some other things interfere with this process? And this is often very similar to what drugs do or what chemicals do in different situations. Okay, so notice here, this was molecule X, and what it's doing, it's actually blocking the ability of the acetylcholine to open that doorway. So the bottom line is we're not going to have contractions if I can't open that doorway, depolarize that motor end plate, and be able to send the signal down and continue with with releasing my calcium, so I can unblock my binding sites for my cross bridges. So this molecule X is actually stopping the acetylcholine from doing what it does by basically being able to bind to the same type of keyhole or receptor site that the acetylcholine would. So when it did that, what we see is it acts like curare or other drugs which will compete with acetylcholine. And so this can either be bad, like curare, which in Amazon's used for the poison and the blowgun darts to paralyze animals, but it could also be used as a medicine or drug to basically paralyze skeletal muscles during surgery to immobilize patients so their muscles aren't moving around. So again, an understanding of this helps us have a much better understanding of what different types of drugs are doing at the cellular and subcellular levels. Okay, another example was what was called molecule Y. And in this case, whereas molecule X was actually blocking the doorway so the acetylcholine couldn't open it, here molecule Y is actually blocking the binding site on the enzyme acetylcholinesterase, which is keeping the acetylcholine from being broken down. Well, if this is keeping the door open and I can't break it down, then I'm definitely going to keep the door open much longer. So what would that be like? Well, this would be like, stigmine and certain drugs that basically, especially for people who have certain diseases that mean they don't have inadequate acetylcholine stimulation. So what that means is if you can keep the acetylcholine that's there from breaking down, then you can, basically amplify the acetylcholine that's there. Now, but just like too much of anything can be too little, or too much of anything can be bad, and large amounts, a drug that would prevent this from being broken down could definitely lead to death, because obviously if this doorway's open, we're going to keep depolarizing, and keep having muscle contractions even to the point of spasms, and spasms could lead to death potentially. Okay, our last example here is molecule Z, which, if you'll notice, it's kind of acting like molecule X. It's actually binding and competing with the acetylcholine, but instead of blocking what acetylcholine's doing, it's actually keeping the door open because it's mimicking acetylcholine. So it's doing the same thing as acetylcholine, even though it's not acetylcholine. So in this case, whatever this molecule Z is doing the same thing as acetylcholine. Therefore, it won't block contractions, it will increase contractions. Well, this is exactly what nicotine does. Nicotine is a acetylcholine mimicker. So when people smoke, it is a stimulant and actually can cause muscle tremors because it is actually mimicking what acetylcholine does. Now, usually from cigarette smoke, even though smoking is bad for you, at that level, it's not going to kill you. But in large doses, again, if you kept those doorways open without being able to break them down, or you kept them open too long, you literally could cause convulsions and potentially death. Okay, so again, when this is nicotine is not hydrolyzed, we know a hydrolysis reaction is where we split water to split a molecule. So if I can't break down that molecule Z and it stays there, it's going to keep the doorway open. Therefore, I'm going to have more muscle contractions. So in a small amount, that's not going to probably affect me too much other than being a stimulant and maybe causing tremors. But as it says, in a large amount, even too much nicotine could cause convulsions and death.